whatever you do, whatever application you do, what you sequence is always a library of DNA fragments. So how are these libraries generated? There are two steps that you need to do to make a library. The first one is you have to fragment your DNA. You have to cut the DNA into pieces. And this is typically done either by sonication or you can also do it by PCR. But in most cases, people use sonication. Then in the next step, when you have fragments, you have to ligate adapters to these fragments. And adapters are short pieces of DNA that you paste to the ends of your fragments. How do you do this? Well, when you make your fragments by sonication, you add your adapters by doing a ligation reaction. When you make your fragments by PCR, well, then the most convenient thing is, of course, to add your adapters to your PCR primers. Adapters are very important for the sequencing. They serve multiple purposes. Adapters are pieces of DNA. As you can see here, they consist of multiple elements, and each element here is colored in a different color. The blue sequences will attach the fragments to the surface on which you are doing the sequencing, typically a flow cell. Flow cells consist either of a glass slide or a surface with tiny wells. And on the surface of the flow cells, there are pieces of DNA covalently attached, and these pieces of DNA can bind to the blue sequences here. And in this way, the blue sequence can fix the fragment on the surface of the flow cell where it can be sequenced. The purple sequences will bind the sequencing primer to initiate the sequencing process. And the black sequences are called sample barcodes, and they allow to sequence multiple samples, multiple libraries at the same time. Every sample will have its own unique barcode, and that means that all the fragments that come from one sample will all have the same barcode sequence. And this means that the black sequence will tell you, hey, this fragment comes from this sample.